Hello, everybody. Welcome to the stream. Welcome to one and all to Biology Trivia Night 3 Extreme Edition. Hello to all of you. The the Stream Elements bot doesn't seem to be working all that well today, but we are adapting our planned Biology Trivia Night stream and upping the ante so that we can raise some money for friend of the channel Psydere, as I will be explaining in just a little bit. Stream Elements bot, I don't know why it won't wake up. Hey Astral, hi, welcome. Member for 2 months. Thank you so much. Yeah. So if Stream Elements bot won't get shifted into gear, I'll just explain how this is going to go down myself. All right. So give me a second to navigate here, and there we go. So yesterday, again, streaming friend Psydere here, psychologist, magical girl, what does she call herself? Magical girl psychologist VTuber, Psydere here, has had some unfortunate unfortunate life circumstances happen quite recently and meets the technical definition of homeless right now. And so, in order to fund a very essential nutrient that British people need in the form of constant consumption of beans, I have modified today's stream to be a fundraiser for her. And so, how this will go down is as follows. For every question that chat gets right, I will donate $8. I'm asking chat 6 questions for a maximum payout of $48. Then I took 13 questions that chat had submitted, and for every one of those questions I get wrong, I will donate $4 for a maximum payout of 52. All in all, if you get all the answers correct and I get all of your questions wrong, I will fork over 100 of my dollars. And for every dollar that chat donates, I will do 2 push-ups at the end of stream, and I will stream as long as it takes to do all of them. I did go to the gym today. I did go to the gym today, so I am feeling a little tender right now, but you know, however long it takes for me to keep stream on, I will finish those push-ups. Is the microphone kind of quiet maybe? On my end it looks okay. I'll maybe be a little closer to the microphone. How's that? I've moved a little closer here. All right. So, why don't we Oh, and by the way, the links to donate to her Ko-Fi. Okay, push-ups it is. Don't actually donate to me, Clockwork. I will put that in and I will forward the money myself. But there are links to donate to her Ko-Fi directly, and that way you actually get the YouTube doesn't take a cut, right? Because YouTube takes a cut when it's delivered to me personally. But if you donate to her Ko-Fi, Ko-Fi takes a much smaller cut. But I will honor the push-ups. I will start off with 4 at the end of stream. All right, so that's how that should work. So there's a link pinned in the chat, and then a link in the description. Yeah, so don't donate to me. YouTube is just going to take a cut, and then Ko-Fi will take a cut on top of that. Donate to her directly, and yeah. And I have the OBS source for her Ko-Fi, so I should get a notification every time someone does use that link successfully. Yeah. So in terms of whose questions are getting asked, I loaded in the so if you've taken a look at my Discord or my community post, there was a form that I linked that compiled all of chat's questions, and I will be doing those questions first because they had submitted theirs in the form that I had linked. All right, but before we get to the chat's question, I am going to ask you all 6 of my own, and I've split them into 2 easy, 2 medium, and 2 hard. 2 easy questions are coming from what you might learn in high school, 2 come from what you might learn in university, and then 2 come from specializations in certain biological fields. So, the dono chat bubble on screen is so cute. The heck, doesn't it make you want to donate? But not to me. donate to the Ko-Fi. Oh, there we go. Mylan contribute 24. Okay. Okay, let's go. Push-up counter 24. That's 48 plus 4. That is going to be 52 push-ups. I should have scaled this completely differently because I will be streaming for a while. Thank you so much, Mylan. Thank you so much donating to the push-up fund here. All right, let's go. Let's get started. So, easy question here, something that you should learn in high school. Which of the following is not an amino acid? Is it isoleucine, tryptophan, fluoxetine, or phenylalanine? Which of these 4? And I will go ahead. Don't answer it. Don't answer in chat right now. Go with the poll here. A, B, C, or D. All right. You can go ahead and answer now, and I will start the timer. Hey, look at me. Look who's got a working timer widget. How I have improved. The production value is so strong here. Ah, there are spoilers. There are spoilers in the chat. What is it? Which one is not an amino acid? Not an amino acid. All right, getting some answers in. Quite a few contributions. 17 votes. 17 votes so far. And it looks like there's some degree of agreement here. All right, we've got 15 seconds left. Last minute for anyone else who wants to participate here. All right. 5, 4, 3, 2, 1. All right. So, oh, the timer runs over. Okay, let's go ahead and stop that. All right. So, chat, did you get this first easy question correct? Let's go ahead and find out. And it looks like we have a rare 100% unanimous agreement here that all 20 of you who had participated agreed that fluoxetine, C, is not an amino acid. And yes, there were spoilers in the chat, but C is in fact Prozac. But doesn't it sound like an amino acid? Congrats! You have all earned a point, and I will be donating at least $8 to the Bean Fund. Let's go ahead and label. That's not right. Let's go ahead and update your score. Where is that? That's here. Okay. All right. 1 so far. Congratulations. Let's see if the next one is as easy as this one. Yeah, fluoxetine, this is Prozac. Prozac is an antidepressant, so not an amino acid. All right. Here is another easy question. All right. Which of the following describes a lipid's electrochemical properties? Amphiphilic, amphiphilic, or biphilic? Which of the following describes? Ooh, 25 to the research funds. All right. That push-up counter. Oh, the scaling. The scaling is quite bad. Oh, crap. I will honor this. I will honor this. It might take me a while, but I will honor this. All right, let's get started on this next question. Let's do A, B, C, and D. All right, go ahead and we'll start the timer. Which of these is not, or which of these describes a lipid's electrochemical properties? Should have seen this at some point in your introduction to biology class, at least molecular biology. Oof. All right, all right. Last half hour stream is going to be just suffering. I don't know if it'll be just the last half hour. Oh gosh. I remember the answer, but in another language than EN, so I may mess up. That is fair. This is very EN centric. All right, that was like 17 years ago. If I have mercy, I mean, you know, take a shot. Take a shot. You can lean on the aggregate chat to converge on the correct answer. Which one of these is the property of the lipid? All right, that is the timer. All right, chat. So what did you say? What did you say? Let's go ahead and end that poll. All right, that is an overwhelming consensus on B. So did chat get this correct? Chat did get this correct. So amphiphilic is one of the 2 acceptable terms. I didn't include the other term here. Describes the lipid's electrochemical properties. The head of a lipid, a molecule, a fatty molecule, is water-loving, and the tails are oil-loving. So amphiphilic refers to the fact that each end of the lipid molecule is loving something else. So one side loves water, the other side loves oil. So chat converged on the correct answer here as well. All right, So what's the other term called? I think it's amphipathic. So it's not quite the same as option A. Not quite the same. All right, congrats, chat. That is 2 questions answered correctly, and I'm going to have to update the correct answer tracker. All right, 2 points for chat. Very good. But that was just the warm up. I was expecting you all to get these right. Now we're going to go into slightly more difficult territory. Gotta push the biology know-how a bit further. So, this is a medium question. Which of the following is not a stop codon? I will not be explaining what a stop codon is. Is it amber? Is it ochre? Is it opal? Or is it ruby? This one's quite a bit trickier. Quite a bit trickier. So I will go ahead and start that timer. All right. Which of these is not a stop codon? Amber, ochre, opal, or ruby? They have girl names? These are precious stones, Phi? They are. They are precious stones. They are precious stones. This is so stressful somehow. It is stressful. This should be not as easy as the easy questions, right? This is a bit more of an esoteric option. Quite esoteric, and not something that I think all classrooms really refer to. But the stop codons do have names, and they do have a theme, which I think is kind of fun. Are these codons or crayons? That's pretty good. Oh boy, I do not remember this. Yeah, it's tough. This one's pretty tough. That's okay, as long as the majority, as long as the dominant answer here, the super majority, no, what is it called? Whatever the most popular answer here is, whatever ranked choice thing, not ranked choice, whatever the most common answer here will be considered for chat's congregate answer. Okay, so that is the timer. Let's see if chat got this question right. All right. So what did chat say here? What did chat say here? Plurality. Thank you so much, Clockwork. All right. So which of the following is not a stop codon? And chat says D, ruby. And that's kind of an interesting... We have Myelin in chat, so this is kind of interesting. If people were purely guessing, there's no way this distribution naturally occurs, right? You all thought that it's like amber is definitely a stop codon, right? Because only 4% of you said it was amber. And the rest seem a little more even, which I think is kind of interesting. The answer is D, ruby. Ruby is not a stop codon. So what is a stop codon? So a stop codon is a sequence of RNA that signals to the ribosome that protein production must end. And so there are a couple of different codes to stop protein production, right? In the DNA, one of these is TAG. And then when you convert that into RNA, it's translated to UAG. And then the name that scientists gave the UAG stop codon is amber. For UAA is called ochre, and then UGA is called opal. Ruby had the right vibes, honestly. I was trying so hard to find a mineral that fit this theme, and I just couldn't. I'm not good at this. Not good at it, but congratulations, chat. That is another correct answer. So when the stop codon is read by the ribosome, it can bind these translation termination factors that break the ribosome apart, stopping protein production. So that is what a stop codon is. Why are they named after minerals? I don't actually know. It's a good question. So let's go ahead and add one more point to chat's score. Really squeezing my wallet here. Okay. Congratulations. Next question. Which of these genetic elements is not always present on an engineered expression plasmid? Again, no answers, no hints here. Thank you for joining, Eisen. Welcome to the macrophage tier of the immune system. Thank you so much for the support. I will honor your membership fee in the push-up counter. I will have to keep in mind that that's $12 so far on top of what I already promised to donate. So let's go ahead and change the push-up counter. That is the $10 reward. So that's an additional 20 push-ups. Oh boy. Okay. Is it antibiotic resistance gene, origin of replication, gene of interest, or promoter? Getting the poll ready, and go ahead. I will start the counter. Which of these is not always present on an engineered expression plasmid? I will explain what that means after chat locks in their answer, of course. There would be no fun otherwise. Thank you so much for joining membership, Synth. Welcome to the Helper T cells. Thank you for being a member of the immune system. I will honor your membership as well. So that is now $17 additionally that I will donate to Psy. That is another 10 push-ups, 32 here. Oh boy. The end of the stream is just going to be a just chatting stream while I do push-ups in between, I think. Yeah. Tough. This is a fun way to teach, isn't it? I think this is a lot of fun. Yeah. An engineered expression plasmid. I'll say one that is useful. I see a lot of good loopholes in the chat. I will say that this is one, this is ready to use. Engineered expression plasmids that are ready to use, I think. This is a medium one too. Oh, hey, look who decided to join us, Stream Elements bot. Okay, so that looks like the timer is done. What did chat say is the answer here? And it looks like we actually have quite a bit of consensus here. Pretty even split between B, C, and D, surprisingly. But most of you said A, which is a lot of fun. All right, so what is the answer? Which of these genetic elements is not always present on an engineered expression plasmid? One that is ready to go here. The answer is A, antibiotic resistance genes. Congratulations, chat. This is so crazy. All right, you've nailed both of the easy questions, and much to my surprise, you nailed both of the medium questions too. So let me go ahead and update that counter. Oh gosh. It's this one for, so what is an expression plasmid? So an expression plasmid is a circular piece of DNA that bacteria can hold onto that is separate from the chromosome. These pieces of DNA often code for useful things like antibiotic resistance genes, or in our case, in the case of the scientist, it can be used to trick bacteria into making proteins of interest. Not just bacteria, but also yeast as well. Anything that can maintain a plasmid can be tricked to do this. And while antibiotic resistance genes are really, really useful to help the bacteria keep that DNA, they don't always have to be there, right? Why is an antibiotic resistance gene useful for a plasmid? Well, it's because if you think about the pressure that a bacteria faces, it needs to be optimized to survive. And it doesn't like carrying extra pieces of DNA around, right? If it doesn't have to keep the DNA, it'll just spit it out, right? It doesn't want to be making things it doesn't have to. But if you give it an antibiotic resistance gene and you expose it to an antibiotic, those factors combined force that bacteria to keep that plasmid. And if it keeps that plasmid, it can be making the protein that you want it to make, right? But the thing is, you don't have to use an antibiotic resistance gene. Of course, there are some applications where you definitely do not want to engineer your bacteria with an antibiotic resistance gene. You can imagine if you are engineering bacteria to maybe clean up the environment, you maybe don't want to give that environmental bacteria an antibiotic resistance gene because that bacteria is just going to teach other bacteria how to fight antibiotics. So what do scientists use instead of antibiotic resistance genes? Well, you have a couple of different options. You have auxotrophic Complementation. Thank you so much for the donation. Oh my god. Oh my god. I should have not scaled this so hard. Thank you so much for the donation. Whoa. Dude, I'm so dead. I'm so dead. Yeah. God, my goodness. It could create chaos or even a disaster if they all have antibiotic resistance genes. Exactly. So it's very useful in a laboratory setting, but you don't want this kind of thing to escape. Auxotrophic complementation is a fancy term for you broke the strain of yeast or bacteria that you're using. It can't make its own tryptophan, for example. And the plasmid that you give this broken yeast can have the gene that allows it to eat tryptophan. So it's the same dependence that you're establishing, but it doesn't rely on antibiotics. It's a lot safer for the environment. Or, and this is my favorite alternative, the toxin antitoxin system, right? This is a system that introduces a poison to the bacteria and also the cure. So if you spit out the plasmid, well, you spit out the plasmid that codes for the cure, and then the leftover poison will kill the bacteria. So it incentivizes the bacteria to keep that plasmid, right? Lots of interesting different strategies for engineering these bacteria. And you can read a lot more about the toxin antitoxin system in my video about bacterial addiction. I like toxin antitoxin because Tat looks like a face. That's so true. That is so true. I am so dunzos. Is the end of the stream just going to be Phi suffering? It is. I haven't done a just chatting stream in a while, so I think it'll be push-ups and then chatting and then push-ups and then chatting. This is. I am going to be so strong. All right. So next medium question. Oh, do I actually have 3 medium questions? I guess I have 3 medium questions. So maybe you can get 7, right? I forgot. Did all offered questions make it in? I excluded 2. I excluded ones that weren't biology related. Yeah. I had enough biology questions to make this only about biology, so I only kept the biology ones here. Okay, so next question. How many amino acids occur in nature? Is it 20, 21, 22, or over 500? Lots of different answers here. So let me go ahead and put that down. Let me reset that timer. Go ahead and catch up on chat a little bit. I love what y'all do, Fine Clockwork. Biochemical and molecular biology has needed communicators on YouTube for so long. I'm glad y'all have taken on that mantle. Thank you so much. I'm just glad to have you all here for the ride. They get rid of the bird one? I don't remember a bird one in the Google form. Bird one? I don't remember seeing a bird one. If we have time at the end, if you submitted one and I didn't put it in and it was a biology question, I'll do it additionally at the end as well. I'm sorry if that didn't end up showing up. I just didn't yeah, I don't remember. Maybe it's in there. I don't quite remember. Occur or identified. To a scientist, that's kind of the same answer, right? Like we can only know what we find out, right? In nature, as in a biological sense, or just naturally occurring. Oh, the Monty Python joke. Yeah, I didn't include the Monty Python joke. I'm sorry. In nature, as in a biological sense, or just naturally occurring. In nature, naturally occurring. Yes. How many? And it looks like we have some disagreement here, but it looks like we do have a majority answer here. All right, so chat, what did you say? We had a bit of a split between the answers, but there is consensus with the over 500 answer. Pretty tricky. This one's a trick question. I like this one. Okay, so how many amino acids actually occur in nature? What is the answer here? The answer is over 500. This is a trick question. If you had taken a biology class before, you may have been tripped up by this because I was very deliberate in my wording. So If you answered 20, that's a very reasonable answer. Very reasonable. There are 20 amino acids coded by standard tRNAs. In the code of in the code of life, right, DNA only has the information to code for 20 amino acids on a normal day. These include serine, proline, lysine, asparagine, etc., etc., etc. These are the amino acids that you should be very familiar with if you've taken any biology class, right? And then there are two weirdo amino acids. So 22 is also not a bad choice. Selenocysteine and pyrrolysine. These are weirdos. There are instances, rare instances, where the stop codon, remember we just learned about the stop codon in my previous question, there are instances, because biology is of course just weird shapes bumping into one another, sometimes those stop codons can actually code for off-brand bootleg tRNAs. I have no idea what these do. These do not come up often in terms of standard biology education, but there are two wacko weirdo tRNAs that do exist that are technically coded in the genome, which is really, really weird. Does this happen often if a ribosome is damaged and can't recognize a stop codon? I don't know. I have no idea. I would love to do a whole stream where I just, for myself, just figure out what these amino acids do, because this never came up in my particular standard education. But what did come up in my education is that amino acids can be modified in nature. Just because there are 20 amino acids that are hard-coded in your DNA, that doesn't actually mean that other amino acids don't exist. Your biology can synthesize additional amino acids. In fact, one of them is actually really, really important, right? Non-standard amino acids that are not hard-coded in the DNA, are also very important, right? Like GABA, which is pictured here, is an inhibitory neurotransmitter. This is a chemical that your neurons release, and it stops its neighboring neuron from firing, right? Which is a part of how your neurons balance firing and not firing, right? You can't be firing all the time. You have to balance when you fire and when you don't. That's how we can actually communicate information. You can think of it as ones and zeros, right? You can't be one all the time. You got to have some zeros in there. So are you also including post-translational modification? I don't actually think that post-translational modification counts as a separate amino acid. I did look this up technically. It has to be some actual different group. They have to maintain the fact that they are amino acids, of course, right? They have to have the amine, and you have to have the acid, right? One end has to be that nitrogen, the other end has to be the carboxylic acid. But I don't actually think that phospho amino acids count. And apart from the phosphos and the other post-translational modifications, there are still over 500 naturally occurring synthesized amino acids. GABA makes a big cameo in the next Clockwork video. Yeah, I saw that you teased that you are doing something in the brain. And as someone who did brain stuff in graduate school, I am very much looking forward to whatever that is. But yeah, so very, very, very cool. All right. Now we get to the first of the, let me update the chat score before I forget. I almost forgot. Almost took away a point from you. Okay, hard mode. Okay, hard mode now. Okay, these require some esoteric biology knowledge. All right, and if you've taken a high school biology class, this might sound like gobbledygook, because this is biology plus going on here. Okay, so which of these refers to a lipid-less membrane-bound organelle? And that might sound like a lot of gobbledygook, but again, I promise you, I will explain everything in due time. Which of these is a lipid-less membrane-bound organelle? And, play fair, don't look these up. Lipid-less membrane-bound organelle. Don't worry too much if you don't know. What is a lipidless membrane-bound organelle? I'm guessing this. Yeah. Play fair. I'm already donating a lot of money. Can I invoke chemistry instead of biology? Well, whatever knowledge base you have, I'm sure, because this is actually the intersection between chemistry and biology, I would say that your chemistry knowledge might come in handy here. Might come in handy here. All right, 8 seconds left. Which of these refers to a lipidless membrane-bound organelle? Oh god, my eyes just glanced over the push-up counter again. Oh god. Oh god. That's a lot of push-ups. Oh, I didn't do chest today. I did back. I did back and biceps today. So hopefully hopefully that'll be okay. Okay, that is it for the timer. So chat, what did you say was the name of the lipidless membrane-bound organelle? And it looks like we have some agreement here, some consensus. So what is chat's consensus? Did that not work? Why did that just disappear? Oh, there we go. It was just slow. It was just slow. Okay, there we go. There we go. Okay. So, which of these refers to a lipidless membrane-bound organelle? The answer is C, biomolecular condensate. C, biomolecular condensate. I'm actually very proud because I think proteinaceous assembly sounds really legit, but that's just a term I totally I totally just made that up. Okay. That's one for me. You know, you've gotten every one of these right so far, and this means I get to keep a little bit more of my money. I'm very, very glad. Condensate, not condensate. I've only heard it as condensate, actually. All right, all right. So, what are biomolecular condensates? Can anybody tell me what this diagram is of? This is something called a Negri body, and does anybody recognize this from a Phi video? Does this ring any bells? Perhaps from one of my most popular, most famous videos? Anybody recognize this thing? Any takers here? Any Negri body enjoyers in chat? Any Negri body enjoyers? Whoa, big big noise from OBS. Neutrophil video? Prion video? Not quite, not quite. Rabies. You are right, Toxo. Rabies. This is from the rabies video. What are these? So what are these? These are viral factories for rabies virus, right? So if you're new to my channel, I made a popular video about the rabies virus discussing why the rabies virus is so hard for the immune system to catch and kill. So if you haven't watched that, please do. It's a great video. Part of their replication strategy is to form these viral factories. These viral factories are completely made of protein. And so this is unusual because before the advent of these high-powered, biology techniques, it was thought that organelles were primarily lipid-bound, that they were covered in a fatty membrane, like the rest of your organelles are, right? Like Golgi bodies and your endoplasmic reticulum, mitochondria, these are all covered in a fatty-like substance called lipids. Biomolecular condensates are getting really, really hot in biology right now because they are additional organelles that are not membrane-bound, and we are just beginning to understand how they work physically, chemically, and biologically, right? So one example of a biomolecular condensate is this Negri body, these viral factories. Very, very, very cool stuff. And if you are looking to get into biochemistry, I highly recommend that you look into biomolecular condensates. And as Clockwork is saying, yes, stress granules are also biomolecular condensates. Lewy bodies, in neurodegenerative diseases are also biomolecular condensates. So many important physiological constructs, items, whatever you want to call it, are really important for the study of health and biology. All right. So, next question. Which of the following proteins anchors the major histocompatibility complex class 1 protein into the membrane? This is a super specific immunology question, and I don't expect you to know the answer unless you are an immunology student. So that is what I mean by hard, right? You don't get this in Biology 101. This is a very, very specialized sort of protein. Again, don't worry too much if you have no idea what this protein does. Just do your best and give me a fair shot. Give me a fair shot at not breaking the bank for the Bean Fund. But yeah, if you've taken immunology, you should be familiar with this. If not, then just do your best. Just do your best. All we can ask for here. Which anchors this to the membrane? Yeah, I just guessed. Yeah, that's totally fine. I love the participation, and it shows me that these hard questions are in fact hard, right? It's no fun if chat just whomps me. I mean, I think you will be, because the questions that chat had put into the Google form are so hard. Like, I think I will be forking over a lot of money in that case. All right, so we have a very even split, which means this is, in fact, a very hard question. Look at how tight this is. And oh, we actually have a perfect tie between B and D. So now I will be generous and I will allow chat to choose between B and D for your final answer. Okay. Get rid of that. One more shot. Let's reset the timer. I will say the correct answer is between B and D. So choose wisely between MAP protein and B2M protein. Raised by a histocompatibility tech, getting disowned if I got it wrong. Oh, Clockwork, you need to get this one right. Anchored into the membrane. Which one anchors it into the membrane? Oh, okay, we're seeing a split here. We're seeing a definite preference. It's an interesting preference, I think. Oh, and now we're seeing things even out. Oh, chat, if you tie again, I'm just going to take the W, right? If you tie again, I'm just going to call this my victory. All right, the timer is about to end here. All right. What is it? What is the protein, this little guy that anchors the MHC1 protein into the membrane? And we'll end the poll right there so that it can't even out anymore. Thank you so much for the gift membership, Synth. Thank you so much. Thank you. I think by default they are the T cell level, right? Correct me if I'm wrong on that. Which should be an additional 10 push-ups. I am so cooked. We are gonna be so strong. All right. So, what is the protein? Yeah, I drew an arrow to it. I did draw an arrow to it. This protein is D, beta 2M, beta microglobulin. This protein is very important to anchor MHC into the membrane. Without it, putting the MHC protein on the membrane gets a lot less stable. So the answer is beta 2M. I have some really good trick answers here, though. You know, I have TAP protein. TAP, if you ever took an immunology class, is the protein. So let me back up. What is MHC? What is MHC? Major histocompatibility complex class 1 protein is a protein that teaches T cells about the status of a cell. If a cell is infected by a virus, for example, as a part of the natural digestive system of the cell, it will chew up some of those viral proteins. Those viral proteins then get loaded into this groove. And let me go ahead and get my pencil out. This groove, right? This little groove over here. And this protein will be shuttled off to the membrane so that the T cell can bind that viral protein. If the T cell finds a viral protein, the T cell will blow the cell up. The protein that loads the peptide, the viral peptide, into this protein is called TAP. So if you had previous immunology experience, that was meant to trick you. That was a very, very tricky question. MAP protein, which people had converged onto, I did, and I love that this was one of the final answers. MAP is just an acronym that you find everywhere in biology. It can stand for like a billion different things, like mitogen activated protein or something. There's another MAP in a totally unrelated pathway. So it just felt like MAP would trick enough biology people, and I love that I was right. I love that people picked MAP because I was like, yeah, MAP is just kind of everywhere and it means 1,000 different things. Thank you so much for the donation, Lindo. Oh gosh, that's another, okay, another 40 push-ups, another 40 push-ups. We're nearing a, a very, very big number here. Boy, oh boy. We're going to be doing a lot of chatting today, I think, as I try to get through this. I might not sleep. I might be here all night. Okay. To get through all this, maybe we should speed things up so I don't have to do any more push-ups. Next question. Oh, let me update the chat. Oh, no. Chat got it wrong. Okay, so I don't have to update this. Okay, next hard question. Production of virulent biofilms is principally governed by which molecular process? Go ahead and start that poll. Is it A, glycolysis, B, binary fission, C, quorum sensing, or D, bacterial persistence? Biofilms. Biofilms are something that I love talking about, and in my upcoming video, I will be talking about biofilms until I am blue in the face. Biofilms are very, very important aspects of bacterial infection, and it is principally governed by one of these processes. I learned this earlier this year. That's so cool. Hopefully you get it right. Love this question. I love biofilms. Biofilms are great. I mean, they're not great. They're great for the bacteria, but they're also fascinating to study for a number of reasons that I will give once we are out of the timer zone. What does chat think? Which of these governs the production of these nasty, scary biofilms? Which of these are the culprit here? Quorum sensing sounds like this kind of silly fake answer I'd come up with on a multiple choice test. I mean, it is a very like ChatGPT set of words. Like if you ask ChatGPT to generate biology technobabble, I that's probably something that they would pick. All right, so what did chat say? What is the principal regulatory thing for biofilms? That was very eloquent. Good job, Vi. What did chat say? Chat is, oh, we have another tie. And this time, yes, the correct answer is in between these two choices. It is in between these two choices. So I will once again give you the opportunity to break this tie. And maybe this time chat will do it correctly. Which of these principally governs the production of these nasty, nasty biofilms? Get the timer up again. Yes, this is how I know I did a good job, right? This is validating that these hard questions end up with a lot more split answers than the medium and easy questions. It is very fascinating. I believe I could probably publish the data on like chat consensus for the easy ones versus medium versus hard. They seem pretty different. I think it's fun. Yeah. What do we think here? Oh, it's close. It's close again. Again, if we get another tie, I'm gonna win. I'm just gonna take the W if we tie. So keep that in mind. All right. What do we have here? What does chat think? A very, very close situation going on here, and that timer is up. So chat, now that the answer has been narrowed down for you, can we get a big consistent answer here? Chat, please, I called us smart so many times already. Well, we'll just see. So chat thinks it's C, quorum sensing. Let's see. Is chat right? Is this the correct answer? Ba-da-ba-ba. See, it is quorum sensing. So what is quorum sensing? I'm so sorry, Mylan. Quorum sensing is the process by which bacteria talk to each other. Right? And this is important because bacteria can either live in a community or they can live in a planktonic state. They can live solo. And when the conditions are right, bacteria need a way to figure out if it is right to make a biofilm. So when would a bacteria want to make a biofilm? Well, if you are the type of bacteria to colonize surfaces, then you would most benefit from making a biofilm. But the problem is that biofilm production is really, really hard and requires the coordination and collaboration of a lot of bacteria. So bacteria need a way to figure out, hey, do we have enough here? Do we have the votes to start a biofilm group? And so quorum sensing refers to the process by which bacteria send signals to each other. And one of those signals is to form a biofilm. Glycolysis, as you should know by now, is the process by which we can take sugar and extract energy from it. Binary fission is the way that many bacteria replicate. They split in half. Of course, I just said quorum sensing. Quorum sensing is the bacterial communication system, and debacterial persistence is a feature of bacteria that enables them to shut down their own metabolism to survive antibiotic treatment, which is different than antibiotic resistance, right? Bacterial persistence, not the same as resistance. Those are very, very different things. Thank you so much, Tyler, for joining the progenitor cells. We'll update the push-up counter to be 284 there. Thank you. All right. Feels like a potential primitive form of multicellularity. Yes, it absolutely is. That's such a clever system. It is. Bacteria are super clever. And this bacteria that I'm doing for my next video is dangerously clever. One of the most deadly bacteria to have ever scarred human history uses biofilms in ways that I could not have even imagined, right? I've kind of teased what this bacteria is in my Discord. I'm not emotionally ready to reveal what it is, but I will tease that this bacteria that I am covering that does biofilms is one that people have been asking for since I did tuberculosis. Since my first real bacteria video on YouTube, people have been asking for this bacteria, and it's coming. It's coming. Be patient. It is coming. This biofilm scar on humanity. All right. Next question. Oh, chat got this right, so I will update the score. Update the score. Okay, very cool. Very, very good. All right. Next. All right. Now we get into the questions that chat had for me. Okay, so I ordered this in terms of easy to hard, although I think a lot of people wanted to see me suffer, which actually kind of works for a stream like this where I'm actively, I'm donating, right? Right. So I don't know if you had caught the Bean Fund energy from Psydere, but the fact that you all had submitted such terrible questions for me, I think works for this stream. Okay, so but we begin with easy questions. Thank you so much for the super chat, Tyler. Don't worry about the push-ups. You'll do enough. Oh, Tyler, I'm a content creator. I can't just take the easy way out. No. I will not take the easy way out. I will add them. But thank you for the consideration. All right. Yeah, I'll do push-ups either until I finish them, which at 300 I highly doubt I can, or if I can't move anymore. If I can't move anymore, that's not going to be very entertaining for anybody. I will be in too much pain to even get up to talking to the microphone again, and I will do the rest of them in a future stream. Don't you worry about that. Okay, so first of the so I owe, let me think. I think I owe an additional $38, I think the counter is. I'll go back, I'll tally them up, but I'm just trying to keep in mind for myself the ballpark estimate. Oh, Toxo, all white knight, some of your push-ups. Fine. Toxo agreed to share the burden. You know what that means, right? That means you can donate more. That means you could donate more, chat. Toxo, it's too late. You agreed. Okay. Sending thoughts in ATP. Thank you so much. Humans have a diploid 2n number of 46. Which of the following statements best predicts the consequences if meiosis did not occur during gametogenesis? So, you have 2n, so 2 copies of your chromosomes during the process of gametogenesis. That double copy gets separated into sperm and egg, which have half of your genetic information, right? So if you did not have meiosis occur, then you would still be at 2n by the time fertilization happens. So if meiosis didn't happen, then the next generation would be doubled. So I'm going to go with the answer of D. You know, silly me, I don't even have the answers ready. Hold on, give me a sec to open the document. That's how I just came out swinging. Let me navigate to this particular question. I need a better way to do this, honestly. This is a bit difficult. Okay, here we go. Okay. So, did I get the answer correct? My answer here is D. My answer here is D. I can take on some. I bet I could do like 3. Oh, you're not letting us guess too? You're right. I should have let chat guess. You're right. I'm getting nervous because the push-up number is so high. So so I'm nervous. Okay, I'll do the first one free. The answer I believe is D. Okay. The answer, D, both parents would contribute 46 chromosomes each, meaning that the number of chromosomes in the offspring would be doubled. So that is a correct answer from me. I don't have to update my counter. You're right, Toxo. You should all guess as well. I'm just freaking out over the very high push-up counter count. Even if I scaled it at 1x, that's still a crazy number. Okay, the next easy question, and this time I'll let chat participate. But chat, you'll only have as much time as I need to answer this question, just to keep things flowing, right? I want to keep on tabbing back to the timer. The mitochondria is A, the only cell whose DNA comes from only the mothers. B, responsible for cellular respiration. C, partly responsible for aging. D, all of the above. Or E, not found in plants. So, my answer here is D. I commend this anonymous contributor for not putting powerhouse of the cell in. That was a very adult thing to do. That is the adult thing to do. Is to oh, Clockwork found a typo. The only cell whose DNA comes from the mother. I mean, what is a cell anyway, Clockwork, right? We all agree that biology is a spectrum, that that definitions and labels are fuzzy. It has its own DNA and it has its own lipid layer. It's just a parasitic cell of sorts, maybe. All right. I will be generous to the submitter here and interpret cell as in organelle. And I will say D, all of the above, right? Mitochondrial DNA comes from the mother. The mitochondria is why you have cellular respiration, and the byproducts, the fact that mitochondria are so good at their jobs at being factories produce a lot of smog and pollution. That's an analogy, it's not literal, but they also produce pollution that can damage your DNA. So I said D, chat looks like chat also said D. What is the answer here? Ooh, I can just download this as a spreadsheet. Maybe that'll be smarter, easier to look forward to. Mitochondria is D, all of the above. I just thought it would be funny because the immediate answer is the powerhouse of the cell, but it's not an option. Mwahahaha. Very devious, Anonymous. Very, very devious. I don't know if it's the only organelle / cell whose DNA comes from only the mother, but that could be adapted. It's obviously responsible for cellular respiration. There's the hypothesis about the mitochondrial basis of aging. Yes, because basically new free radical oxygen species are generated during respiration, causing you to age. Yeah, and mitochondria are found in plants. They don't just have chloroplasts, but they also are found in plants as well. I really wanted to say that it helped us discover the origin of life on Earth, but Ugu, no space. Yeah, yeah. Fantastic, easy level question. A nice little warm up here. Do I have a video in symbiogenesis? That's a bit out of my expertise. I do not have one yet, although I think that is something that I would love to look into. Okay. So, next easy question. An autoimmune condition is identified that results in the destruction of Schwann cells. How does this condition impact the lives of patients? Oh, sorry, I forgot to show. Chat did, in fact, pick D. So, Schwann cells. This is a question about Schwann cells. I thought the mitochondria was primitively a different cell that was phagocytosed. Yeah, but you would call that something else, right? You would call that whatever thing it was before. You wouldn't call it a mitochondria because a mitochondria is the thing that exists today, right? Let me go ahead and look at the answers here. They are unable to regulate their cell cycle and develop cancer at a higher rate. They are unable to myelinate their neuronal axons, and nerve signaling is slow and inconsistent. They cannot produce helper T cells that are unable to mount an effective immune response. And D, their gametes cannot undergo recombination and their offspring will be less genetically variant. So every year I don't actively study neuroscience, I feel like I lose a lot of it year to year. But I believe Schwann cells are the cells responsible for myelinating neurons. So I'm going to go with B. I'm locking in my answer as B. Chat, what did you think? Chat also says B. Wait, plants don't have mitochondria? They do. They have mitochondria. Yeah. So yeah. Chat said B. I said B. What is the answer here? Let's see. This one is B. Yes. Schwann cells myelinate neuronal axons that don't have a role in those other things. Yes. Okay. Very, very good. So far, my money is safe where it is. But don't you worry, Chad. I've taken a peek at some of these other questions, and they are difficult, right? I think this is the last of the easy questions. All right, next up here. Medium. Thank you, Petal, for this one. Which chromosome is associated with Patau's syndrome? Oh, I'm not. I'm only updating mine when I get one wrong. That way I can quickly calculate how much money I owe. Sorry, I should have made that clear. I have gotten none wrong so far. I will only update when I get them wrong. So, which chromosome is associated with Patau syndrome? So let me get this ready for chat. I can already tell you right now, I'm guessing. I don't know what Patau syndrome is. I don't know why this is in medium. I don't know what this is. I like the vibe of B. So I'm gonna go with B. That's just a complete and utter guess. Because I have to put it in an answer. And I'm going to continue to stare at my second monitor until chat has a little bit more time to lock in an answer, but I'm not going to wait that long because I myself am just completely guessing. So what did chat think? Chat said C. Okay. And by quite a large margin too. So chat and I disagree. So what is the answer here? All right, the question from Petal. The answer is C. Damn. Sorry. C. Patau syndrome is also known as trisomy 13, where there is an extra copy of chromosome 13. This is similar to Down syndrome that has an extra chromosome on 21. Okay, yeah, there's no way that I wasn't guessing for that one. So that is, I'll do -1 so that it is more clear that I am counting my losses, my L's here. Can we post an answer in chat? I'm ignoring chat when I'm answering, and there's a poll, so when I put the poll up, you can contribute. Okay. Next question. This one's from Matthew, another medium question. Another medium question that I don't think is a medium question, by the way. I'm gonna go ahead and start the poll. The lack of this tumor suppressor gene causes Li-Fraumeni syndrome. What is the name of the gene known as the guardian of the genome? Is it CDKN1A, TP53, ERBB, or CMYC? And so, chat, go ahead and lock in your answer. I'm going to answer. P53 is a very prominent cancer gene, but I also know that MYC is another master regulator of the cell cycle and is also heavily involved in the development of cancer. The question comes down to which of those two do I believe is given the moniker guardian of the genome? I want to say P53 because that is like the thing. I actually don't know if TP53 is something different than P53. I'm just guessing that that's an extra letter. And if that's if that changes, if TP53 is different from P53, I might have been screwed there. But yeah, for me it is between B and D. But I think I'm going to lock in B as my answer. P53 with an additional leading T for some reason. And it looks like chat agrees with me because there is agreement. But if the answer is D because TP53 isn't a thing, I'm going to be so mad. I'm going to be so mad. So what is the answer here? This is the question by Matthew. The answer is B. TP53 encodes the okay. TP53 encodes the protein P53, which has many anti-cancer properties like the induction of senescence, which is when cells stop dividing, DNA repair, and apoptosis. Okay, so TP53 is the gene, P53 is the protein. That is what I am guessing. I see. Okay, well, okay. Thankfully, I got that one correct. So that is all right. That is all right with me. I'm not too mad about that one. I should have known because TP53 is all capitalized and the P53, that's the protein, is not capitalized. And that's how you can kind of tell when something is referring to a gene or a protein. So that should have been a dead giveaway. Okay, next question. I'm doing surprisingly well. All right, this one's from Pierce. Thank you, Pierce. A person suffering from moderate to severe hypothermia may suddenly start to feel very hot and get the urge to remove their clothes. What is this phenomenon called? So I actually believe I know which of the four I would narrow it down to, which is another way of saying I have it in between two. So the answers provided are pseudo-hyperthermia, paradoxical hyperthermia, paradoxical undressing, and hypothermia-induced psychosis. So I believe with a pretty high degree of confidence that it is one of the paradox answers, and this is very devious. Paradoxical hyperthermia or paradoxical undressing. If I were to go with what makes more sense in English, I would say that it's probably C, paradoxical undressing, because paradoxical is modifying undressing. It is a paradox that you are hyperthermic, but you are undressing, whereas paradoxical hyperthermia doesn't quite make as much sense because that is implying that the hyperthermia is a paradox and not the act of undressing itself, which is a lot of gobbledygook to say that I believe it is C. I'm locking C in. Chat, what did you think? Chat says it's also C, but not by a large margin, which makes me kind of worried. Oh, I know this one. Life is good. Yeah, this one is fun trivia, and I'm gonna be embarrassed if I get this one wrong. So, Pierce, what is the answer here? The answer is C, paradoxical undressing. Nice. Why isn't the answer showing up? Sorry, 1 second. Why isn't the explanation showing up? I mean, is it? Okay, there we go. Paradoxical undressing is a behavior that can occur during hypothermia due to either damage to the hypothalamus associated with body temperature regulation or vasoconstriction failing in the peripherals due to muscle fatigue, causing an excess of blood to rush into the limbs. A person will suddenly feel very hot and get the urge to undress in order for them to feel colder. However, this makes the situation worse as they are still suffering from hypothermia in reality. I like this question. This one's good. This one's a nice bit of fun biology trivia. Very, very good. And also, kind of tough. the polling is really, really late, but I think that one was an appropriately medium question for chat. All right, next one. Medium. This one's from Martin. Which of the following is the largest contributor to the differences in melting temperature between AT and GC rich DNA molecules? This one I'm very confident I know the answer to. And if I get it wrong, I'll donate double. If I get it wrong, I'll donate double. Yeah, so I'm pretty sure I know this. And yeah, if I'm wrong, then I will donate. I will -2 from this because I am that confident. But as I'm reading these again, I'm getting less confident. Yeah. So the answers are A, electrostatic interaction, B, hydrogen bonding, C, pi stacking interactions, and D, covalent bonds. I'm not looking at chat. I'm locking in my answer, but I'm giving chat time because I don't need time to think. I've already locked it in. All right, chat. I'm locking in my answer right now. My answer is going to be C, pi stacking interactions. Pi stacking interactions, C. Chat says B, hydrogen bonds. See, that is what I believe to be the trick answer. I believe that is the trick answer. Melting temperature between AT and GC rich DNA molecules. Oh, maybe I'm wrong. No. I locked in already. I'll say if I get this wrong, I may have misread the question. Maybe I was too confident. I'll still eat the -2 if I get this wrong, of course. Okay, so chat locked in B. Oh, no, I think chat's right. Oh, I saw the answer I liked the most and I picked it right away. This is going to hurt. Oh, I should have read it more carefully. Okay, Martin. Largest contributor. Oh. The answer is C. Contrary to popular belief, GC-rich DNA molecules have higher TM values due to more favorable base stacking interaction that occurs with repeated GC pairs. The difference in the number of hydrogen bonds is only a minor contributor. Considering denaturing typically occurs in aqueous media, there are also ample hydrogen bond donors and acceptors in the local microenvironment. Okay. Oh, thank goodness. I came out very strong, and as I waited, I got more and more. I got more and more worried. Okay, so this is gonna require some drawing. So let me make this real big, and let me draw some very simple DNA ladder. So this is my very simple DNA ladder. And chat picked hydrogen bonding, this answer, which is not a bad answer because that describes these bonds, right? Bonds that hold this part of the ladder together, right? They kind of are glue between the two strands. But what pi stacking refers to is this interaction, the stability that occurs on this plane. So not this plane, right? But the up and down forces. Right, and the more, the larger, because here's the thing: hydrogen bonds, they aren't quite as additive in when it comes to biomolecular stability than pi stacking is. So because DNA is such a huge molecule, this force, the force of just this huge mass that generates all these van der Waals forces, is going to be additive because DNA is such a huge molecule. Whereas hydrogen bonds, that's only holding together these individual rungs, right? These rungs are what is being held together in hydrogen bonding. But pi stacking occurs here and scales off of DNA size. I don't know like all the chemistry/physics behind why this is the correct answer, but it is the one that I have internalized. But don't they have the same number of aromatic rings? I believe it is the interaction of the pi bond, which you're gonna have to consult a chemist over. But that is what I remember vaguely. A biologist explaining chemistry. But doesn't the melting temperature refer to when 50% of the DNA is double stranded? How is C involved at all? van der Waals forces can stretch over very, very large spaces. So you can think of van der Waals as being, it's not really gravity, right? It's not really gravity, but it scales with how big a molecule is. And so the larger a molecule is, the more van der Waals forces it is going to have. And I believe pi stacking is an example of van der Waals forces because you have a larger total electron cloud, and van der Waals forces is, oh, wait. Am I getting van der Waals? Chemistry. This is why in my neutrophil video I go chemistry, because I am so bad at chemistry. I'm not gonna butcher this any further. I believe the answer is pi stacking, but I'm actually not good enough to give you more of an explanation than Martin had already gave. And of course, if I end up being wrong on this, I will correct it in the stream description so that I do not propagate misinformation. Okay, I'm doing surprisingly well here. Okay, next question. I thought pi stacking is typically aromatic ring stacking. Okay, I will make a note. Somebody make a note in my Discord to double check on the veracity of this question here. I will donate -2 if it is not pi stacking, because I would have picked pi stacking no matter what. So someone in Discord, just in like general, you can tag me to double check the melting temperature thing. Yeah, keep me honest here. Yeah, van der Waals scales with surface area. Oh, thank you, not size. Thank you. It's been very long since I had chemistry. Okay, yeah, check me on that, because I don't want to speak that confidently of a field I don't fully understand. Okay, next question. Let me go ahead and get this loaded up here. Thank you, oxidized salmon cream. Why can't neurons replicate? Bonus points if you can explain the molecular mechanism. A, differentiation disrupts conductive paths. Oh, excuse me. B, replication would activate both BDNF/TRKB pathways. C, replication disrupts critical synaptic connections. Or D, neurons upon differentiating engage in G0 permanently. I have to be honest, I wasn't super confident because of my answer here, because I don't I'm very weak in cell biology. Even though I did neuron stuff, right? I think cell biology I've always been weaker. And again, every year I don't do neuron stuff as my primary job, I feel myself losing my grip on my understanding of neurons. But I believe I will lock in D, and I cannot explain the molecular mechanism. I do not remember. I knew it at one point, but I do not remember. So D is going to be my answer here. Chat says, I think chat is tied, actually. Is chat tied? We'll find out like 5 minutes because the polling is so behind. Yeah, my answer is D, which looked like one of the dominant answers that chat was suggesting. Okay. So my answer is D. What is the answer? Oxidized salmon cream. The answer is, oh, there are 2 correct answers apparently. D is correct because neurons lack regulatory proteins, checkpoints for DNA replication and cell division. And the reason why that is, they say, is B. Replication would activate BDNF/TRKB pathways. Very interesting. Okay, so I feel like I'm just scraping by the skin of my teeth, but I'm doing quite well here. Wait, olfactory neurons replicate, don't they? Yeah, I got in trouble for implying that they don't in the rabies video. And I was like, I knew that, but I figured I wanted to simplify it for an audience. But like from the rabies video, I learned that my audience is actually very scientifically mature, and I can add that nuance in, right? Not all neurons are post-mitotic. Oh, well, let me back up. I don't want to say that. I don't think that's correct. Not all neurons, not all populations of neurons are non-renewing. That is more correct, because you first have to have a neural stem cell, and those can differentiate into a proper neuron as they migrate towards certain niches. I believe that's how that works. Correct me if I'm wrong. I'm pretty sure, Guy, you did like You did neuron stuff. They don't replicate. Stem cells do a differentiation. Yes, 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 yes. I caught myself instantly. I was like, that doesn't sound right. Neurons don't do mitosis, but they differentiate from stem cells. Okay, very, very cool. All right. So I'm holding strong at a -1, but we have yet to even get into the hard question, which is very, very scary. Okay. Hard. This one's from Dylan. Oh gosh. This one is gobbledygook right off the bat here, right? This is immediately gobbledygook. And there are 5 answers. I like, you don't have to participate in this particular one because I can't, like, I'll do this with 5 answers, but yeah, I forgot to mention in the forum that YouTube's polling can only handle 4 options, which is kind of annoying and silly. But I will answer it like it is 5, and if the answer is E, just don't answer. I think that's a fair way to do it. I think not fair, not totally good, but you know. So Dylan asks, what best describes canonical row utilization RUT sites in E. coli? Oh, C pore with regularly interspersed G's, G pore with regularly interspersed C's, A pore with regularly interspersed G's, AT rich with no discernible pattern, GC rich with no discernible pattern. Okay, so row, I believe, is something to do with translation. Or maybe not, because if it's translation, it would be a U and not a T. So maybe I don't know what row is. Row utilization sites in E. coli. Okay, so my guess is going to be, God, I don't even know. Row, what is row? I'm a mammal biology guy, so even though I do bacteria for the channel, I actually don't have a great microbiome foundation. I'm going to go with I'm going to go with one of the first 3 because AT rich and GC rich, I'm just going to say that that's going to change the melting temperature of that local area a bit too much for it to feel naturally occurring. So I'm going to go with C. I'm going to lock in C as my answer. A pore with regularly interspersed G's. I have no idea what row utilization is in E. coli. If it's not something to do with translation, protein translation, that is. Okay, so chat said D. And poll. Yeah, I said C. So Dylan, what is the answer here? We got 1 out of 5 shot here. The answer is not immediately obvious from the submitter. Okay, so let's read the explanation. The bacterial transcription terminate. Oh, transcription. It's not translation. Okay, so it's not having to do with protein, but having to do with making RNA. The bacterial transcription termination factor Rho binds specific sequences in nascent RNA to facilitate transcription termination during Rho-dependent Okay, so it is termination. It stops something. I just got it wrong. It's not protein, it's RNA. During Rho-dependent termination, in E. coli, Rho is generally considered to be essential to cell viability. Rho binding sites, known as RUT sites, are typically cytosine-rich and guanine-poor regions in the RNA. It is hypothesized that these cytosine-rich sequences promote efficient Rho loading and stabilize its multimeric structures. So I'm guessing that answer is cytosine-rich and guanine-poor. Is that even one of the options here? Cytosine-rich and guanine-poor. Oh yes, B. So that's probably B. Okay, that's B then. Okay, so if you answered B, congratulations, chat. Very, very smart, chat. I certainly did not remember. So I'm going to take that hit right now. B, that is B. So -2 for me. Yeah, there's no way I get that. There's no way I organically get that. I think I learned it in my first year of graduate school because we did a whole brutal semester of bacterial biology, but I have not applied it since then. All right, next chat question. Oh, why are Down syndrome and Alzheimer's dementia often comorbid? Okay, let's go ahead and get the answers up here. Why are Down syndrome and Alzheimer's dementia often comorbid? So if you don't know what comorbid means, it means often occurring together. So A, abnormal hypo. Okay, sorry. For the let me do the previous question. Let me do the explanation. So remember how earlier in stream we talked about translation termination? How like there's a specific RNA sequence that the ribosome reads and then it disassembles the ribosome. This is basically that, but for RNA polymerase. So it stops transcription. It stops the production of mRNA. And apparently there's a part of the DNA. This should have been obvious to me. It's AT, so it's a DNA thing. But whatever. I wouldn't have gotten it right anyway. But yeah, this has to do with the sequence on the DNA that tells the RNA polymerase to bugger off and disassociate. Okay, so C. Abnormal so under phosphorylation of tau results in neurofibrillary tangles. There's no obvious connection to Down syndrome in that one. ApoE4 is upregulated in Down syndrome. That is plausible. C. Amyloid precursor protein is affected due to being on chromosome 21. Also plausible. D. Presenilin 1 and 2 are both commonly mutated as well. I don't believe there are linkage risk factors for Down syndrome. So I'm eliminating D because I don't have a reason to believe why someone with Down syndrome would be at a statistically higher risk of having a Presenilin 1 or 2 mutation. So that one I'm eliminating. A, I'm eliminating for no obvious connection to Down syndrome, so it comes through B or C. I think C is a very, very plausible answer if I believed amyloid precursor protein is the etiological agent for Alzheimer's dementia, right? Because that is actually a hypothesis that is falling out of favor. Now, has it fell so much out of favor that I don't pick that as the answer? I think so. I think I go with B. I think I go with what I think is correct instead of what I think is what this answer is looking for. So I'm locking in B. So what does chat say? Chat overwhelmingly going with C, which is, the answer that I would go with if I weren't trying to be more clever than this question implies, which is possible. It's possible that the obvious answer is the correct one. So Anon, what is the right answer here? Okay, so this one is Down syndrome. Okay. Ooh. All right. C is correct. Trisomy 21, so Chad, you are right, causes overexpression of the amyloid precursor protein and thus increased beta amyloid deposition. I would argue that right now, because of all the controversy surrounding amyloid beta, that is maybe a correlate. I don't know enough about Alzheimer's to fully fight back on that, but biology is always evolving. A is wrong because hyperphosphorylation of tau leads to yeah, that makes sense. Hyperphosphorylation of tau leads to neurofibrillary tangles and it has nothing to do with Down syndrome. Yep. B and D are wrong because ApoE4, Presenilin 1 and 2 are not affected by Down syndrome. Because they're all on different chromosomes. Yeah. Okay, I got that wrong. I tried being too clever, and chat, you have bested me in this question. Well done, chat. Well done. All right, next question. Here we go. Okay, this one's from Jacob. What is the rarest symptom of malaria, specifically Plasmodium falciparum? I've got a higher chance of just guessing this correctly because that is what I will be doing. Why is my computer so slow right now? That is a little annoying. It is being slow. Let me actually try reloading chat because maybe it's chat that is specifically being bogged down. So give me a second. You can ponder the answer here. Although I am personally not doing all that much pondering. This to me is yeah, this is gobbledygook to me. Just loading up the stream. I want to reopen the chat because the chat tab is just so laggy. Like the polls just come out so slow. Reopen chat. Oh my gosh, an ad. Watching an ad on my own stream. Um, whoa. Okay. Pop out chat. All right. Hopefully, with the chat reloaded, this will go faster. Okay. Black water fever, vomiting, acute pancreatitis. So, I believe Plasmodium falciparum makes its way into the liver of people. So, GI stuff makes sense to me. So, vomiting makes sense to me. Fever also makes sense to me. I don't know if Plasmodium invades the pancreas, so I'm going to go with C as my answer, right? C. I'm locking in C. So, chat is also saying C. Chat is also saying C. Okay. So, what is the answer here, Jacob? I will check here. Where is your question? What is with this thing where it's not? Okay, here it is. Here we go. The answer is A, Blackwater fever. Blackwater fever is the answer. It is historically the rarest. It can only happen if you get malaria multiple times and use quinine, a very old anti-malaria drug. So it looks like you are in chat. You want to talk a little bit more about what Blackwater fever is? And I will update my L counter here. I thought I was being a little too, maybe I was a little under clever, right? I should have been like, Blackwater fever sounds too specific. Sounds too specific. Yeah, I thought maybe pancreatitis because I think falciparum is liver, but there's no reason why that can't spread to pancreas stuff. And I have no idea what Blackwater fever is. So that is very, very interesting. Okay. Being liberated from my hard-earned money. We will continue. All right. Oh my gosh, another row question. What are the odds? Are these two different people? I think these are two different people. Did I get two different row questions from two different people? Well, the benefit of this is that I know what this is now. I think I know what this is now. So this one has 5 answers, so I don't think it's worth making chat do this particular poll just because it's complicated with 5. So I'll read this out loud. Translocase, homoheptamer, ATPase, transcription termination factor, or helicase. So this is the thing that binds DNA. We know that it stops mRNA from being made. So D is correct in the terms of like B is true. So it can't be the correct answer. Translocase, these usually refer to enzymes that move things. Sure, maybe, I don't know. Homoheptamer is a description of the structure. Homo being all same and heptamer being 7 subunits. So is rho a 7 subunit protein? Could be. There's no obvious reason to eliminate that as an answer. C, ATPase. Well, it is an enzyme, and ATP is often used to power enzymatic reactions. So I'm leaving ATPase as well, and then helicase, which is an enzyme that unwinds DNA. Because this is a transcription termination factor. But does it also have helicase functionality? Or is this a different row? That's the thing about biology, is that there's no checking to see if previous names have been used before. So that's always an option as well. I think I'm going to go with helicase because I don't see how a transcription termination factor would need to unwind DNA. Yeah, let's just commit. I'm gonna go with E. E for helicase. That is my logic here. Okay, Evan, what is the answer? Evan? Okay, the answer is uh okay, let me read this out loud first because something's not making. Oh, okay. The answer is B. Homoheptamer. Rho is not a homoheptamer, as in 7 units. Rho is a homohexamer. A homohexamer. So instead of being 7 repeating units of the same thing, it is 6 repeating units of the same thing. Oh, that's so. That is devious. That is devious. That is devious. Really, bro? Yeah. Evan, you got me with that one. That functions as a transcription termination factor. Rho-mediated transcription termination, also called extrinsic termination, is one of 2 methods that bacteria uses to stop transcription. Rho is essential for most gram-negative bacteria and some other bacteria of clinical importance, such as tuberculosis. However, most gram-positive bacteria can survive without Rho, although growth defects are commonly observed in Rho mutants. Bicyclomycin is an antibiotic that works by inhibiting Rho. Very, very cool. I did not. There's unfortunately, I don't think there's an explanation for the helicase part of this, right? Because why does it need to be a helicase to terminate transcription? That doesn't make sense to me, but that's okay. I will trust you, Evan, and I will take the L here. Update that score, -5. So I told you, y'all ask really, really tough questions, really, really hard questions here. Helicase makes sense to you? It doesn't make any sense to me. Maybe I'm being silly. Are there different subtypes or isoforms? I'm not sure. I'll go to sleep now. Long day tomorrow. Thanks for the stream. Thank you for coming. Thank you so much. All right, next question. This one's from Violet. What is the most abundant epitranscriptomic modification in human mRNA? Another 5 answer ones, so I'll just do this one. A, pseudouridine. Helicase gets terminated by the G-rich region. But why is it a helicase? I don't quite understand. Like, the helicase portion pops off when it hits the G-rich region? That's why it... Oh, is it? But yeah, I don't quite understand. I think it's interesting. I do want to look it up, or if you can spare some additional elaboration, that'd be pretty cool. Okay, yeah. So the most abundant epitranscriptomic modification in human messenger RNA, is it A, pseudouridine, B, inosine, C, N6-methyladenosine M6A, 5-methylcysteine, cytosine, sorry, or E, none of the above? Ooh. This one is hard. And I'm going off of an educated guess here. Because I remember M6A being a thing I learned about. Oh, wait. Is M6A a microRNA thing or an mRNA thing? Oh, whoa. The most... Oh, I should know this. This is something I should know. I should know this. Damn. Oh, gosh. I'm going to lose my license that I don't have because you don't need a license to teach at a university level. I'm going to lose my street cred, man. M6A, is that microRNA or messenger RNA? Oh, and if it's not M6A, what else could it be? Methyl something or another? It has the word methyl in it. Maybe it's still M6A. Is it methyl? Or is it just plain methyl? Does M6A refer to just plain methylated mRNA? I'm going to go with C. I still think it's M6A. It just feels right. Thank you so much for the donation. That is 20. Please correct me if I'm wrong. The font is very small right now. Is that 21? 21? If it's 21, I'm going to assume it's 21. Please correct me in the chat if I'm wrong. I'm going to add 42 to the push-up counter. Thank you so much. Thank you so much. Okay, very, very good. Very, very good. Okay. Okay, yeah, I'm gonna go with C, M6A. So Violet, what is the right answer here? Oh, okay. So the answer here is in fact C. Thank goodness. Okay, so N6-methyladenosine is the most abundant and well-characterized so let me back up. An epitranscriptomic modification is a modification that happens to mRNA after it's been made. Okay, and that can modify the translation of the mRNA, can modify a whole bunch of other things as well. Yeah, there's all sorts of variation. And this is exactly what a hard question should be. It should be something extremely niche like this. A well-characterized epitranscriptomic modification, human mRNA. M6A regulates mRNA localization, stability, degradation, and translation efficiency. Epitranscriptomic modifications are chemical modifications of RNA nucleotides analogous to the chemical modifications of DNA studied in epigenetics, i.e., cytosine methylation. Enzymes that deposit these modifications are called writers. Oh, I didn't know that. That's cool. Proteins that recognize modified RNAs are called readers. Let me back that up and say that correctly. Enzymes that deposit these modifications, these M6As, are called writers, and then the proteins that recognize these modified RNAs are called readers. And enzymes that remove these modifications are called erasers. That's very cute. That's another amber, opal, ochre kind of triad that I think is really, really fun. That's cool. That's awesome. I love that. These are so niche. They are. That's why they're hard, and that's why I've taken L's on multiple ones of these questions. Y'all really, really challenged my biology know-how. I joined to see 5 with -5. Yeah, that's -5. I will donate I think $4 for every minus I have here. So that is 20 so far. But I think this is the last question. Yep, that is the end of the presentation. Great trivia night, guys. I thought that was a lot of fun. What's less fun is what I'm going to have to do now, which is push-ups. Okay, so we'll do this in a couple rounds because there's no physical way I can do all of these at once. And if you want to challenge yourself, a bit of a shared fitness challenge maybe, why don't you do these with me? I'm going to start with, I think I'm going to start with 20. I'm going to start with 20. I'm going to come back and then I'm going to do some just chatting. And we'll do this again until I can no longer do a push-up or I finish them. But I'm gonna be real. Given the number on screen, I don't think I'll be finishing these. I genuinely don't think that finishing this is in the cards for tonight. Let me just say. But thank you so much, y'all. This has been really, really fun. Boy. Here we go. Okay. Okay, so push-up time. All right, 20 to start. Okay, 20 to start. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. All right. That is 20 down, y'all. Let me update that push-up counter. Oh, okay. That wasn't bad. That wasn't bad. I have been going to the gym, so before when we took our vacation in Paris, when we got back, I was not doing very well in terms of physical fitness, but I'm retrieving some of that. I'm retrieving some of that, and now I will start a timer because I can't just put off the next set of push-ups entirely. So we'll do like a Pomodoro, but from hell. Let's do 5 minutes of chatting. How are you all doing tonight? Tell me about what's been going on, chat. We're deep into the school semester for a lot of people at this point. So tell me how your semesters have been going. And if you're a student, tell me if you're taking biology, tell me how that's going too. Yeah, and in the meantime, I kind of want to see. Yeah. Yeah. especially because since I no longer teach, I'm itching to hear some of how things have been going with all of you, just because I miss teaching in a classroom. It's kind of lonely making content by myself in this room, you know? And I want to hear what's been going on in universities at large or high schools at large, right? I do know that I saw a couple people say that they are uh taking A levels or taking their first biology, their first like big biology class. I think that's very, very interesting. There's no such thing as rest, there's only emails. I was chatting with another streamer in their chat about emails as well. Mylan, how many times do you check the email address of what you're about to send before you click send? I feel like most of my time back when I was teaching in university was spent double checking that I had the correct email address. I just never, because here's the thing, if you teach long enough, you'll eventually get students with duplicate names that have, exact same first name, exact same last name, but they have different email addresses. And I was always so paranoid of sending like a final project grade to the wrong student. I'm mostly sending replies, but if I'm sending the first email, always. But like, how many times? How many times? How deep does your paranoia get? Mine goes pretty deep. I will. The funny thing about like grading some of those final projects is that the biggest spike of anxiety usually came when I had to actually send out the grades. Like grading itself wasn't that stressful, but like the 20 minutes of like very carefully ensuring that everything is packaged correctly, I think I don't miss that part. Once, check it again in the email and the roster. Oh, okay. So you actually go back to the roster. Um, once is efficient. I'll put it that way. I'm crazy in the sense that I check all the time. Speaking of encroaching anxiety, I'm seeing the timer tick down for the next batch of push-ups, and if I only do 20 at a time, I don't think I will make headway very quick. So next round, I'll do 30 of them. Do any of you have any guesses as to what the next pathogen is, by the way? I think if we see a correct answer, I might reveal it here. Did you ever mess up and send grains to the wrong person? I personally never have, but I have been close because I had forgotten that two of my students shared the exact same name. Yeah. And my memory is really bad, right? Like, unless I really study something, there's no way I can remember it. And, part of that was displayed fully here because, Mr. Biology here with fancy degrees from fancy universities, I still got a whole bunch of these wrong, even though I learned about row before. I'm almost certain of it. So I only ever do grades to the course site, so it's not an issue. Oh, that's messed. That's so convenient. No, we were still emailing students their grades because we couldn't do it through the well, we had to email them grade sheets, right? Like feedback for their projects, and that can only be done through email. Well, we couldn't do that through the course website. There are Discord hints for the next pathogen. There are some hints in the Discord. Exclamation point Discord if you haven't joined already. Let's do that. Yeah, so if you go to the channel upcoming videos, you can see, if you just like scroll up, I gave some hints as to what that next pathogen is. We're discouraged that because email is not too secure. See, that's smart. That's just straight up smart, I think. That just makes a lot of sense to me. I don't know. I was thinking S. aureus or P. aeruginosa due to biofilm formation. Those are really good guesses, honestly. And those guesses would be really good if all you knew was that I was doing something dangerous and did biofilms. Those two guesses would definitely be the first thing that I would think of, right? P. aeruginosa and the S. aureus, but it is neither one of those. All right, the timer's gone. I'm gonna aim for 30 this time. I'm gonna aim for 30 this time. Oh my goodness. I'm gonna try not to use my Toxo Assist. I'm gonna try not to use it. But here we go. Another 30 I'm going to aim for. 30. 1, 2, 3, 4, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Oh, oh boy. Oh, I'm tired. 21, 22, 23, 24, 25, 26. Okay, 26. I think that's good. That's good enough for now. We can end on an even number for the next round, right? Oh my goodness. Yeah, it's been a while since I've done push-ups too. Should count to 300. A nice even number, easy to keep track. Math is easy. Oh, oh yeah, I'm doing all right. I'm doing all right. Um, the first 20 felt fine. The next 26 is rough. I would have had trouble even doing half the scaling. But yeah. Yersinia pestis. It also forms biofilms. Oh, very, very, very interesting. Interesting that. Very, very interesting indeed. Right. Oh god, I'm never gonna finish these if I take full 5-minute breaks. So let's do a short 3-minute break here. Oh boy. Yeah. You would think that like doing content full time would allow me to actually participate in pop culture, right? Because everyone and their mother is playing Silk Song these days. And I have been grinding at this dumb video in almost all of my free time. Uh because I just don't want to get this out so badly. And watching everybody play the game, I'm like, I am on the periphery of culture. By the time I play Silk Song, nobody will care about it. I've been good at avoiding spoilers, but is anyone here playing Silk Song? I've also been looking at like the keep an eye out on Legends of Arceus as well because I like looking at new megas. I don't even play Pokemon anymore, but there's something very compelling about designs for Pokemon because I think this is maybe a huge supposition, but I think Pokemon was probably pretty important for a lot of biologists, right? Little cute animal friends that love you, and of course have interesting biology if you really want to apply it that way. And even though I don't play the games anymore, I always like seeing what Nintendo does. Has anyone seen Mega Victory Bell or Mega Malamar? And there's a third one too. Gosh, who's the third Mega? Mega Malamar, Mega Victory Bell. I always Mega Halucha. Yeah. Mega Halucha. I love these designs. Mega Dragonite. Oh, is that one out too? I didn't know. Is Dragonite coming up in Legends of Arceus or is it already out? Yeah, Halucha. I like the Halucha Mega. I think Pokemon Company is becoming very, very comfortable with making these goofy ass designs. Ooh, so less than say so word there. These goofy designs in Mega Victory Bell. And I think at first, these kinds of designs really I did not like, but I've warmed up to this goofy side of Pokemon, right? Things like Mega Executor, for example, right? Infinite Neck. Um, it's going to be in ZA and has already appeared in the anime. Oh, Mega Dragonite. I remember seeing designs. I don't know if I've seen canonical designs, but oh, I want a Mega for my favorite starter. I heard discourse that Mega Meganium might be a thing, and if Mega Meganium were a thing, and I liked the design, I would maybe consider playing Pokemon again because I love Meganium. I think it's a great Pokemon design. It's not my favorite Pokemon, but it's up there. Ooh, okay. 3 minutes. 3 minutes done. Time for the next set of push-ups. This time, I'm not going to aim too much higher. I think I'm going to attempt 20. I'm going to attempt 20 here. All right. Okay. 20 push-ups. Be right back. 14, 15, 16, 17, 18, 19, 20. I'm sweating. Okay. 20 more. I'm gonna maybe try to do 100. Try to do 100 today. That might be the goal, because 360, say 300 what? I don't know. Words. Oh, I'm blushing. That is accurate. I'll just leave that on because it's accurate. I'm definitely feeling the blood pumping. The best design wise is Mega Malamar. I agree. I don't really think that Mega Halucha is all that different from regular Halucha. Uh, and the Mega Victory Bell is just, it's hard to look at. Hey, chat, how is everyone doing? I just got here. You got here at a good time? Question mark. I am suffering. I agreed to do 2 push-ups for every $1 donated to Psydere's Bean Fund, and chat was feeling quite generous tonight, much to my detriment. So even though the trivia part is over, there's still the suffering part, which I am doing right now. Yeah, but I think the problem with being YouTube full-time is that I have less life to talk about, which is kind of a weird thing, right? I don't get out to experience things that happen to me, and so everything that I chat about are like vivid hallucinations I have within my own head. Which isn't a bad thing. I suppose there are certain topics that I think I like talking about that eventually become members only videos to a certain extent, which I don't mind sharing. I think those are fun topics of discussion. But chat, while I am recovering from doing push-ups, do you think there is a way to enjoy food incorrectly? Would you say that there's a way to enjoy food incorrectly? Like a steak or pho or something like that. What do you think about that, chat? Can you enjoy food wrong? Is there a wrong way to prepare and enjoy food? Whatever that means to you. Don't ask me, well, what do you mean by right or wrong? How does that strike you? How does that hit your ears when I say, is there a way to enjoy food incorrectly? Eating it with a straw? You know, I would agree. I would agree. Piffy says yes. So what would you describe? How would you describe eating food incorrectly? I mean, if it's actually unsafe, like eating raw chicken, safety is a little hard to quantify, right? It's a little hard to quantify. There's like food safe foods, but like where you put what's a good example here? Foods that are a little risky, I'll put it that way, like moonshine. It's not controlled. You know, you could have a batch with methanol in it, could make you go blind, but is that wrong to enjoy? Is it wrong? Eating mashed potatoes with a fork? What about that would you describe as wrong? That's kind of what I've been thinking about this. I occasionally go back to this idea because I think the wrongness is what's kind of appealing here. Uncooked food is no bueno. Other than that, no. That is interesting. So you would not say, for example, that eating $80 ribeye with ketchup is wrong. An overcooked, super well done ribeye with a side of ketchup. And that is how you order it, not how you ended up salvaging it, but how you ordered it. You wouldn't call that wrong. Eating it in a way that misses the intended flavor of the preparer. Yeah. See, I think that is a reasonable category, a reasonable distinction, a reasonable definition of food rightness and food wrongness. Enjoying it by removing what makes it enjoyable, like getting a milkshake at a restaurant just to take it home and turning it into not a milkshake. That is interesting too. But there's this theme of intention. You guys debate about pineapple on pizza. Yeah, that's another hot internet debate topic. Depends on whether you mean wrong as in incorrect or morally reprehensible. Let's stay away from morally reprehensible. That's one definition that I'll just say categorically that's not what I'm talking about. Like, I'm talking in the category of like pineapple on pizza, if there's something wrong with that, or ketchup with steak, etc., etc. Is that something that you think about? And do you think you can be wrong on food preference? And while you think about that, chat, I am going to do another. I will try another 20 push-ups. I did have a slightly longer than usual break here. I will be right back with another 20 push-ups. 6, 7, 8, 9. Oh, these are getting difficult. 10, 11, 12, 13, 14, 15, 17, 18, 19, 20. Oh God. My chest is feeling like jelly. Okay. It's another 20. I think we're not too far from doing a full 100 tonight. I look forward to that. I think there's no wrong way to enjoy food. It's just that amount of enjoyment you get varies on what you do. Okay, would you say that there is a suboptimal way to enjoy food? Does that strike you as something that is more acceptable to say, right? Is there less and more optimal ways of enjoying food? What do you think about that? Oh, speaking of food, little Perkinji over here is wondering when she is going to be fed. Perkinji, you want to say hello to chat? You want to say hello to chat? No, you're just going to be silent? I feel like the fact that you're saying enjoying implies it's optimal. Well, I mean, there's spectra within enjoyment, right? You can like something and you can love something. So I don't really know if it is optimal per se or implies perfect optimization. But if you were to, like, yeah, I wouldn't think too hard about it. I mean, everyone's tastes are different. You can optimize that, but the conditions are going to vary between people. There is no wrong flavor you can enjoy. It is only when you deface the food itself, when the food is no longer what it was or is habilitated by your actions. I think that's interesting. I'll go off of that. I'll just read Misa's comment first. The only exception would be if someone prepared food very specifically for you to eat as they made it, in my opinion, like a professional chef or a friend cooking you their own food from their account. Yeah, so I think that is where I have been thinking in that particular realm of when somebody else makes food for you. Because I think what the okay, so I don't know if I have to like recap this discourse, but like if you say that you enjoy pineapple on pizza, there will be very vocal camps telling you that you are wrong for doing so. Or if you say that you like your steaks well done, or if you like them with ketchup, or if you drown your pho in hoisin sauce or sriracha, there are people online and in real life who will judge you for it, who will say that this is the wrong way, this is not the intended way of eating a certain dish. And I think because of the fuzziness of the English language, that we cannot agree on whether or not there should be a right and wrong, and to what degree it actually matters, like what the stakes are. Stakes, that's double entendre for you, right? Because yes, this is all internet bluster, and in some ways, it doesn't matter, right? Who cares about the way that you eat certain food? If you enjoy ketchup with your $80 well done ribeye, if you're not hurting anybody, why does it matter? Why does it matter? Who cares? And I think that's true. I think it doesn't matter how you enjoy your food, except for the context that Mies had pointed out, right? There's something different about eating food that somebody else prepared for you. Because food that they prepared is a gift, and a chef matters less than someone else cooking for you, right? A chef, you pay them to do whatever. And as soon as money is exchanged for their labor, who cares what you do with your product, right? It's a gift that you can't return. You shouldn't return, right? But even so, there's still an element in me that says you have to acknowledge that eating a ribeye well done is wrong. It is not the correct way to eat the ribeye, right? And I think that both of these camps that say, oh, there is a right and wrong, and the camp that says, it doesn't matter, eat it however you want, I think we both have to come together in the perfect middle and acknowledge that you should not eat a well done steak, a well done ribeye with ketchup. You just shouldn't. Why is that? Because you are cooking out dimension of certain foods, right? You paid a lot of money for a certain experience, and by asking for this to be prepared suboptimally, you are shrinking the area under the curve of the integral of your enjoyment, right? It is just worth less after you've treated it a certain way. Now, that might sound like I am pro food judgment, but I'm actually not. I think there are two things that should happen with regards to this discourse. One, we acknowledge that there is a canonical way, you could say, to enjoy food, but also we acknowledge when we depart from canon, because everybody does. It makes us human, right? I don't have every conventional food opinion in the book. I have weird things that I eat too. And I will admit fully that I am reducing the integral of experience when it comes to my weird food preferences. And everybody has them. So I think both of these fierce Romeo and Juliet family, wars that are going on, the pro food convention people and the food anarchists can come together, and we can agree that there is canon, that they were built on logical principles, but also acknowledge that each individual is allowed to usurp those expectations, but they must be honest about it. Like, the person eating well done ribeye with ketchup doesn't get to say that their way is correct, right? Does that make sense? Does that idea kind of pass, right? Because I don't exclude myself from this comment, right? I too, like, the thing that I think that upsets most people is that, I like pho, right? When I get the bowl of pho, I always drink a little bit of the broth to show appreciation to the person who cooked it. Like, I will drink the thing you labored 18 hours for, right? 18-hour bone broth or whatever, days of bone broth in some cases. But then I drown it with hoisin and sriracha almost every single time. I don't think I've ever had pho that I liked more than pho plus sriracha and hoisin sauce. And there are camps that say that's completely reasonable, that's completely respectful. Adjust it to your liking. But then there's a camp that says you should enjoy pho as intended and not add hoisin and sriracha into it. That you are compressing, you are destroying the layers of intricate aromatics and the labor that went in, and just reducing it to a salty, sweet broth that can be made for cheaper. And I acknowledge that. And I know that North and South Vietnam have different opinions with regards to pho purism. And I don't know who comes out on top, or if this debate is raging, right? But what I will say is that I myself acknowledge that there are fewer things going on in the food taste experience when I drown pho with sriracha and hoisin. But I don't care, because I prefer that experience. But I can also say that this is my personal preference, because I prefer my food to be very strongly flavored. And let's be real, pho broth is not the most flavorful of options. I'm not saying it is not delicious, because it is delicious. But in terms of like the absolute number of neurons firing in your mouth, it's just not as much as some strongly flavored things, right? It's delicious, but it could taste more. Not better, it could taste more. And you can make it taste more by adding a bunch of stuff to it, which again, I think is wrong, but I do it anyway. And by acknowledging that, I think it says something, right? It says something. So I'm going to read chat, and then I'm going to do my next round of push-ups. But I just want to leave you with that kind of thought. And I want to hear what your reactions are to that idea of like embracing the fact that convention exists, but also being honest about when you subvert it. Yeah. I guess then that goes into food for sustenance versus food for art, and you can mess up your sustenance if you want, but messing up someone else's art is different. That is true. That is, I agree with that. I messed up that chef's art anyway, but I enjoyed it in the original context first before I destroyed it. People can get judgy when it comes to out of the norm things. Give mac and cheese and mustard. Some people can find it weird, but it's genuinely good. Same for like pregnancy cravings. Yeah. I acknowledge that too. Like, mac and cheese with mustard is not a conventional pairing, but I can acknowledge the possibility that it is kind of good. Integral of enjoyment. I think a lot about integrals for someone who's not very good at math. I love eating mustard seed on bread and nobody can stop me. Yeah, dude, I'm not shaming anybody for weird food preferences, right? I'm not shaming. I have weirder ones that I'll talk about after the push-ups. But this is more to be like, how can we bridge the, the rhetoric is so hot, the divisions are so deep. How do we bring people together? It's acknowledge the food standard, but also I am an individual with a personality and weird, and my personality is weird. Food personality is weird. Showing this to my friend next time they judge my unconventional food tastes. Yeah, I mean, maybe I'll edit down to a short so it's easier for them. I put sriracha on anything. I like pineapple on pizza. I also find pineapple on pizza acceptable. I don't even really understand why people find it offensive because sweet and salty go together in so many other contexts as well. Your opinion is wrong. It's only when you put a ribeye in a salad that it becomes a problem. Yeah. Everyone has one of these. I used to not eat pineapple pizza because I had reaction to pineapple, but when I grew out of it, it wasn't that bad. It's not that bad. I don't think so. Pineapple and jalapenos on pizza is such a god tier combo. It sounds like a great combo. Oh, you'd make me hungry, and I have so many more push-ups to do. I'm gonna go have another round of push-ups. Hopefully, I would really love to end the night on a nice 200. I don't know if that'll be possible, but for now, another 20 push-ups. Be right back. Oh my god. Yo, my form is breaking down, yo. My form is decaying. It is going, but I think we're pretty much at 100 push-ups done so far. Yeah, this is tough. Y'all donated way too much money. Bye, Sandy and Butter. Bye-bye. Is this considered the wrong way to enjoy food? My sister peels her McDonald's chicken nuggets. That's That's crazy. So what I will say is that is an unconventional way to enjoy food, and I say more power to her. More power to her, but she can't pretend that that's normal. That's all I'm asking is that the way forward, the way to unite all of us is that she cannot admit that that is normal. Okay, so why do I think that? Why do I think that there is something important about acknowledging when your food opinions are weird, right? And I, this is not novel. People do this all the time. I think it is because this is something that is a social identifier. I don't know if Mylan's still in chat, but you can critique my amateur psychology, amateur sociology hypothesis here, right? Like, I think that your taste in food is an identifier that other people can use to either loop you into their in-group or exclude you into an out-group, right? This is a metric of division, which is no good, right? You don't want to be excluded from a group of people, feels bad, but people do it anyway, right? We already talked about how food opinions can be a line of division. People can get very judgy and shut you out for having bad food opinions or food opinions that don't align with their own, right? And that's unfortunate, right? We don't like to be judged. We don't like to be pitted against one another or thought lesser of, right? People do get very judgy. They think lesser of you for enjoying things this way. And what I say to that is when you embrace the weirdness, but you also acknowledge the weirdness, I think it brings people together, or it has the potential to. Because let me put it this way, like, you shouldn't judge people for how they enjoy food. For the most part, all right? I think there is room for judgment, and this is going to get me canceled. There is room, there is a reasonable there is a reasonable way to judge people for their food opinions. And let me make the case here. And you can push back on me. I might change my mind if this brushes up against you in a prickly way, right? I think you can judge people by their food preferences, but only when they cook for you or they recommend a restaurant to you. I think that's appropriate. Do you give Mr. I cook ribeyes to Well Done and enjoy them with ketchup? The grill, when you are in a backyard cookout, right? When you're at a backyard barbecue, do you give the reins of the cooking implements to the guy that likes his steak well done? I think it's okay to be like, hey guy, maybe you take a break, right? Maybe you go do something else and let somebody else cook for the group, because conventional food tastes will, by definition, have a wider, safer appeal to more people. And I think that is an okay context to judge people's food tastes in. You shouldn't think of them as lesser, right? But I think you go, from a practical perspective, I don't trust you to do this one thing because you haven't made it clear that your mouth is not wired in the same way as mine is. And that's not a moral or ethical judgment. It is simply making a decision based off of learned data. So I think that food weirdness, when you acknowledge, like, for example, say I'm the guy that likes well done steak with ketchup, and I make it clear that I know that this is wrong, people might trust my ability to cook other things because I acknowledge that my understanding of conventional taste is still intact for other things, right? Maybe shoe leather steak guy can make a mean salad, right? It's more dimensional that way. This debate has lost the plot. I think so too. But I still think there's something here, and I want to refine it for a members only video at some point. Like, there's something like in between. It's okay to judge people for their food choices, and you shouldn't judge people at all for their food choices. There's something in the middle that I think is reasonable, and that is something that I'm trying to work out. Does that kind of make sense, chat? Or is this all gobbledygook, right? Do you think that it is unconditional, that you should not judge people for how they enjoy food in every context? Or is there like a base case, like an edge case where that is acceptable? Or do you trust the guy that likes his steak well done with the grill? Maybe you are less judgmental than me. Oh, here we go, Mylan. Like, on one level, I think you have a coherent train of thought, and on the other, I think it is not a train that I am permitted as a passenger on. Why do you say that? That's interesting. I think it is not a train that I am permitted as a passenger on. Why not? I think that's kind of interesting. Or perhaps it's too spicy. Perhaps the idea, I cannot follow these tracks. Okay, let me rephrase this. Is there ever a situation where it is okay to judge people based on their tastes in food? Is there a morally, ethically acceptable situation where food judgment is okay, apart from preferring dishes that are actively cruel, right? I'm not going to define what cruel is because that is a whole nother thing. But like with the steak example, like, do you think it is acceptable to judge and act upon judgment when you know someone who likes well done steaks to prevent them from being the communal cook for your group of friends? As a picky eater who grew up with a highly sensitive stomach, I will never judge. I am being judged. Oh. That's an interesting perspective to have. Would you say, as a picky eater who grew up with a highly sensitive stomach, I will never judge. I am being judged. What's a food that you... Maybe I don't want to get too deep into it. Maybe it's something that I need to sit on a bit longer. Maybe I need to cook this idea up. It's not fully baked in my brain yet. Let me go ahead and do another 20 push-ups. Oh, it's getting late. I should wrap up stream soon, but I want to get down to 200. I don't want to carry over more than 200 push-ups to the next stream. So I will be back. I will do another 21 of these push-ups. Oh, oh gosh. It's another 20. Yeah. Okay. One more round of chatting, and then I will do another 20, and then that'll be the end for today. Yes, the communal cook should be able to cook steaks that everyone would enjoy. Yeah, so I think like the transparency when you discuss food preferences. I think acknowledging that convention is a useful tool to think about, but also doesn't define oneself, but also has some practical implications as well, as in like communal cooking or restaurant recommendations. Incredibly minor, not that important. Me appearing to destroy 5 member video ideas. Insert tuxedo mask. My job is done here. Meme. Yeah, Mylan crushed another one of my ideas for members only videos earlier this week. We have a shared Discord, and if I feel like my ideas aren't cooked, I'll just offer them up in the Theorem Discord. And yeah, earlier this week, like Mylan brought up some very good points about that topic that caused me to take pause and think of a different idea while I let those ideas percolate in my head. Basically, that video idea was about why the song Golden from K-pop Demon Hunters was more emotionally resonant with me compared to Let It Go from Frozen. Because I think Golden and Let It Go have identical roles in their respective movies, but Golden is the one that like emotionally gets to me. And I had this whole Google document, I had a table, I had categories and analysis across different dimensions of each song. And then Mylan like pointed some very good things about the context in which Golden is sang in. And I was like, oh my god, I have to like actually refine this idea to something that actually makes sense. You should talk about the gut microbiome and its connection to your cravings in your next video. That is difficult because I'm not a microbiome guy. I think for me, the microbiome is a tough video subject. I think it's tough because there is so much hype around it. And so in order to do a good job, I need to be practically measured, right? Because I hear, even amongst people that I like, a lot of hope for microbiome stuff that I personally don't see as being supported by good data, put it that way. Not to say that I don't think the microbiome is important. I think it's incredibly important, but just I'm always careful when it comes to talking about the microbiome because I feel like people treat it like a panacea, like, oh, everything is the microbiome. And maybe that's true, but I am cautious. I will only talk about the microbiome when I am sure there is something real to talk about instead of just correlations. And that's really hard to do by the nature of the microbiome, right? I wouldn't say that I'm a microbiome skeptic. My graduate school was big into the microbiome. I love reading about the microbiome. However, I feel like if I am going to be contributing to the narrative that the microbiome is the secret to the biological universe, I want to be able to cite something that I feel very comfortable with, which is kind of hard because for me, I am most comfortable in the concrete, right? We talked about the spectrum between concrete and abstract in the schizophrenia video. Like psychiatry for schizophrenia, that's the most abstract understanding of schizophrenia. That's not any less legitimate, right? schizophrenia is a complicated disease and psychiatry treats the person and people are very complicated. Me, when I read about schizophrenia, it is often at the level of molecules and cells, very concrete things that don't have an obvious connection to a person's identity, personhood, etc. So in that way, I feel very similarly to the microbiome, where there are big, exciting things that people are saying about the abstract connection the microbiome has to health and biology. But for me, I'm less comfortable with drawing that connection because I feel more at home in the concrete cells, molecules, and stuff. I hope that is not too disappointing. It's not stopped me from talking about the microbiome before. I even did a paper with Mylan as a collaboration a while back, and I did a whole video about the immune system within the gut. But those I felt more like I was able to hone or refine the exact things that I wanted to say, because I don't believe in overhyping the microbiome. I think we have to be very careful because this is my personal thing, but a lot of people take advantage of people's general belief that the microbiome is the way forward, selling all sorts of nonsense, right? And I don't want to contribute to that mysticism, is what I'll say. Yeah, I get the hesitation. It feels like a lot of it is hype or people exploiting the current scientific knowledge in the field to make claims to make money. Yeah, I've seen respected researchers go to the dark side and sell nutritional supplements that don't have double-blind controls to show efficacy, right? It is a lot of tempting money that people are looking to cash out on. And it's disappointing when it happens, right? It's sad. You can never say that they don't work, right? You don't know that these supplements don't work because they're usually not tested. Not with any sort of acceptable rigor to be called medicine, of course. So that's kind of the attitude that I would like to impress upon you all with regard to the microbiome. It's very exciting. It's probably the answer to a lot of biology, but those answers don't exist quite yet, except for in rare clinical cases, things like fecal microbiome transplants for infant IBS or something like that. Like there's a very particular set of conditions where microbiome treatment has been shown to work. And outside of those, it is taking a nutrition supplement person's word for the effects, right? Outside of that rigorous clinical testing, it's all kind of low-end mouse stuff. And that's, yeah, I don't want to make it seem more than it is. That's a long-winded rant, but I think I'm just procrastinating this last 20 push-ups, which I will do, and then I will call it a stream from there. How's that? Last 20 push-ups, my form already degrading to the point of no return, but I do feel very, very tired. So wish me luck, wish me luck. These last 20 push-ups. Be right back. Oh God. 2, 6, 7, 10, 11, 12. Oh my goodness. 14. Oh my God. 15, 16, 17, 18. Oh my God, this is really the last set. 19, 20. Oh my goodness. Holy cow. Okay, that is another 20 push-ups. Putting the counter at 200. I will aim to do the next 100 in the next stream. Thank you all for hanging out with me as I put myself through physical pain and for contributing to the Psydere Bean Fund. I hope you all have a wonderful rest of your day, and I will see you next week. What am I doing next week? I will see you next week when I am exploring Science Hell with Mylan. I collab with Mylan where we explore the deadly sins of the science world. Thank you all so much for coming. See you next time. Bye, y'all. Bye-bye.